So, hi students, welcome to another episode of Microbiology Decoded. So, today we are going to study about one of the important technique that we are using in the microbiology lab, that is the gram staining technique. Have you ever wondered how this microbiologist can identify quickly different types of bacteria using the microscope? So, this is one of the simplest and powerful method around the world, that is the gram staining process. So, let's get into the video. So coming to the introduction, the gram staining is a laboratory technique used to differentiate bacterial species into two groups based on their cell wall. So this is the important sentence in the gram staining: differentiate bacterial species into two groups based on their cell wall. Bacteria are of two types, that is gram-positive and gram-negative bacteria. Gram-positive bacteria will appear as violet color and gram-negative bacteria will appear as a pink color. Okay. Gram-positive bacteria, which contain a thick peptidoglycan layer, and gram-negative bacteria contain very thin peptidoglycan layer. This is the fundamental thing which differentiate them based on their color. Okay. So the staining is developed by a Danish bacteriologist named Hans Christian Gram in 1884. So this is the introduction. So let's check what are the stains we are used in this technique. So coming to the procedure, we need a smear. Okay, we need to heat fix it. It contains maybe even gram-positive bacteria as well as the gram-negative bacteria. So before staining, the bacteria must be glued to the slide, and heat fixing will denature the bacterial proteins and ensure the specimen doesn't wash away during the multiple washing process in the staining procedure. Okay, after the smear is done, we will start using different kind of stain. So many kind. I think four kind of stains I'm using here. So let's get into the staining technique. And first we are using the primary stain, that is the crystal violet. We will keep that for around 1 minute. So the primary stain is crystal violet and we will keep that for 1 minute. It is the basic dye which enters the cell wall and the cytoplasm of both the gram-positive and gram-negative bacteria, and after washing it stains everything in a purple color. So after the primary stain process, whatever in the slide, it will look like a violet color, even if it's gram negative and even if it's gram positive bacteria. Okay. So after the primary stain process, we are going to use the mordant. Here we are using as a grams iodine as the mordant, and we will pour over the slide for 1 minute, and the slide is rinsed with water. Okay, and this grams iodine is acting as a trapper layer. What it actually does is it's bind with the crystal violet to form a large crystal violet iodine complexes, and that are too big to easily escape through the mesh-like peptidoglycan layer of the cell wall. Okay, and this iodine form what it complexes with the crystal violet and make it difficult for the stain to wash out. So that is the purpose of iodine here. And coming to the decolorization process, we are using acetone or alcohol as a decolorizer, and we'll keep that for at least, not keeping, we'll wash that for at least for 2 seconds, only for 2 seconds. Okay, and this is the most crucial and time-taking and time-sensitive step in this gram staining process. In the case of gram-positive bacteria, that it has a very thick peptidoglycan layer. As I said already, gram-positive bacteria cell wall contain a thick peptidoglycan layer. So, because of its thickness, the alcohol can't penetrate into the cell wall and it will not decolorize and take away or wash out that primary color that it's already stained. Okay, in case of gram-negative bacteria, the alcohol dissolves the outer lipid membrane. So, this gram-negative bacteria contain extra outer lipid membrane, that is a lipid membrane, and it leaves a hole in the thin peptidoglycan layer. So, the outer lipid membrane dissolves and it leaves a hole in the thin peptidoglycan layer and allow this purple dye to wash out. So it will enter the peptidoglycan layer and it take away the purple color and they become colorless. So that is what happening in the decolorization process. That's why we said like it is the most crucial technique in this gram staining process. And the most common mistake in lab is over decolorization. Okay, if you leave the alcohol for more than 2 seconds, even the gram-positive bacteria will lose their purple color, and it's leading to a false negative bacteria. So don't keep the decolorizer for more than 2 seconds. Okay, that is a very crucial step in this gram staining technique. And the last stain we are going to use is the counter stain. So we are using saffron as the counter stain here. It's added for 30 seconds. Okay, since the gram-negative cells are now invisible, saffron is added to the stain and give them a pink color. Gram-positive cell remain purple because it contains already the primary stain. Okay, so the invisible cells are now, that is the gram-negative cell, will take up the counter stain, that is the safranin, and it will appear as a pink color. So that is what happening after adding safranin. Okay, and after this all staining procedure, we need to keep that slide for air drying, and after drying we need to add some oil and observe them under the 100x, that is the oil immersion. So here is a demonstration by using a picture. So here we can see first we added what? Crystal violet. So even the gram positive and gram negative cell appeared as a violet color. Then we added the grams iodine and it will form a crystal violet and iodine complex. And next we are adding the most crucial thing, that is step, that is the decolorizer step. So acetone we can use here, and it will take up the color of the gram negative and it appears colorless, and the gram positive bacteria will appear will not wash out their color. And coming to the last step, that is the counter stain, we are using here safranin, and the colorless gram negative bacteria will take up the counter stain, that is the safranin, and it will appear as a pink color. So the gram-positive bacteria with thick peptidoglycan cell wall retain the crystal violet stain and appear as a purple. And gram-negative bacteria with thin peptidoglycan cell wall loses the crystal violet and when treated with the alcohol. So because of their cell wall composition, gram-positive and gram-negative, because of their cell wall composition, it take up a different dye and appear in a different color. Okay. So this is what happening in the gram staining process, and that's how they are getting a different color. So let's check the principle. Organism which resist decolorization by organic solvent and retain the primary color appear violet are called gram positive, whereas the organism that gets decolorized and take up the counter stain will appear as a pink are called gram negative stain. So this is the principle behind the gram staining technique. So that's all about the gram staining. I hope this video helped you clearly understand the principal procedure and interpretation of the gram staining. If you found this video helpful, please like, share and subscribe to this channel for more microbiology content. And thank you for watching, and I will see you on the next video.